Good morning to all of you. Today I have to discuss about the root pressure experiment, how we can see the root pressure and plasmolysis. So for the root pressure experiment, what we have to do, we have to take a herbaceous plant in a pot, and then what we have to do, stem, you have to cut the stem a few centimeters just above the soil, okay, and then above the base you have to cut it a few centimeters. You have to cut the stem above the base of the soil of the root, and then you have to just attach it with a manometer. So manometer is there, which is an instrument used for measure the small pressures. Okay, that is called manometer. You have to just insert a manometer in the cut portion of the stem. So after few hours, what you will observe in the manometer, the mercury level is rising, and it is due to the generally the root pressure. So what happens when the stem is cut, the water which is present in the xylem, that is coming out, and it is just filling in the manometer due to the root pressure. So what we have observed, that simply the root pressure is generally responsible for carrying the water up in the xylem. So in the xylem, when the water will be there, it is only due to root pressure. So this is simple experiment which you can read also. So this is clear, root pressure. Next topic is your plasmolysis. So in case of plasmolysis, what is happening? Whenever the cell is placed in hypertonic solution. So in case of hypertonic solution, what will happen? The cell will shrink. Okay. So in exosmosis, so the process will be exosmosis. When exosmosis will take place, what will happen? The cell will shrink, and the plasmolysis, protoplasm, in this case the protoplasm shrinks and is pulled away from the cell wall. So there will be a gap between the cell wall and the protoplasm. Already we have seen in the turgidity condition. In case of turgid condition, what happens? There is a little space, and every cell in cell there is too much water. Water cannot be absorbed more. That condition is known as turgidity. When there is minimum amount of water absorption, then it is called as turgid condition. So it is just like a flaccid condition. So here it is the protoplasm is pulled away from the cell wall, and the space between the protoplasm and cell wall is covered or filled by the external solution. Whatever we have taken external solution, hypertonic solution, so that solution will be filled inside this. So that will be shrinking of the cell. So when this case will occur, initially it is known as incipient plasmolysis. So this is called as plasmolysis. This process is known as plasmolysis, and the cell will be called as plasmolyzed cell. Okay, initially that plasmolysis is known as incipient plasmolysis, but if it is continuing, since the plasmolysis is, if the plasmolysis is continuing, then it will be known as permanent plasmolysis. So if it is incipient plasmolysis, when you will again put it inside the hypotonic solution, so due to endosmosis, what will happen? The cell can regain its size when it is incipient plasmolysis. But if it is permanently plasmolyzed, whenever you will keep the cell in the hypotonic solution also, the cell will not recover. Okay, because it is permanently plasmolyzed. So if incipient plasmolysis has taken place, then only when you will transfer the solution or the cell from the hypertonic solution to the hypotonic solution, the cell will regain its original size. But in case of permanent plasmolysis, you will not be able to see this type of condition. So generally plasmolysis takes place in the living cells. Most of the cases it is taking place in the living cells, but in some of the cases you can observe also there osmosis, endosmosis, exosmosis takes place. But generally for the living cell, sometimes in most of the cases, 99% of the cases in living cell, plasmolysis takes place. So I think it is clear to all of you. Plasmolysis, very simple topic, that exosmosis and endosmosis. Exosmosis, that is the coming out of the water in the when it is cell is placed in the hypertonic solution. So it is when it is placed in the hypertonic solution, outside concentration is low. When outside concentration is low, it means water content is less. That's why the water is coming out of the cell. It is exosmosis. So in the next topic we will discuss about the root pressure theory and how the active and passive absorption is responsible for the absorbing water and minerals. Till then, thank you.